Okay, so our next speaker is Rachel Adams from University of California at Berkeley.
Okay, good morning, everyone. I'm happy to talk to you guys today about work that I've been doing as a group at Berkeley, where we have looked at taken different biological, engineering, and sort of chemical-based approaches to looking at sources of microbes indoors. So first I'll talk a little bit about let's see. Okay, so first I'll talk a little bit about background, the work that sort of has motivated the questions that we're looking at now. So we first started doing some population level surveys about what are some microbial exposures that people experience in residential and classroom environments. And what we see is that as a generalization, most of the fungi are coming from outdoors, and this is true for bacteria, but laden on that you see a source of humans. And one of the things that was really important was that we sort of new technological advances have enabled high technical replication. And this is going to be really important in questions moving forward, because as we've heard, a lot of these questions are not going to be answered by identifying one or two additional microbes, but looking at how subtle shifts in overall community structure are driving some of these observations. And detecting those differences are going to be enabled with these new technologies. So I mentioned that humans are a source, particularly for bacteria, but also with fungi. So we did experimental work in a chamber where we could control the number of people and their activity level, and there were two sort of dominant processes related to occupancy that were particularly important. One was the resuspension of the reservoir microbes that were in the space that the people were occupying. Another was this reminder that humans are transport vectors themselves, often through their clothing, for microbes that they're picking up in other places. And so this sort of work got us thinking about the nature of the material and the source. So for example, floor dust is both a source and a sink for airborne microbes. And so, and those are sort of different exposure routes and have a different impact depending on the target population, for example, children during infancy or adults. And so on some level, it's kind of like what Shelley was mentioning, a lot of this is about particle dynamics and particle movement, of which microbial-derived particles are one subset of that. But on another level, I mean, microbes could have the potential to be living, have the potential to potentially be a live, intact organism, and none of these methods that we were using were getting at that. So then we started to wonder about what is alive? And like a lot of good questions in life, there's this conundrum has been touched upon in a Monty Python skit. So the famous dead parrot skit, there's the pet shop owner insists that this parrot is not dead, when it clearly is, that it's merely resting. So, so while this is clearly a dead parrot, what is this and what is it doing? So modern microbiology is a lot of looking at DNA sequences on a computer screen, which doesn't give us any information about sort of the biological state of this organism. And on some level, it may not matter if the organism is alive, for example, as a trigger or as an environmental trigger. But if we want to understand sort of the function of these microbes indoors, we do need to have a sense of whether they're alive or capable of becoming alive, or capable of some growing if conditions change. So one approach that we've really looked at is the use of RNA. And so we looked at the biomarker signatures when you're targeting DNA and you were targeting RNA. And we specifically went to dry and wet areas of the home, thinking that in wet areas, most of the microbes there are going to be happy because they have both substrate and water to live, and in dry areas, it was mostly going to be organisms that settled there and were clinging to life before they died. And so what we found overall is that the microbes on these wet and dry areas of the home were not similar, which makes sense, but that and when you're looking at RNA and DNA, there's a lot of overlap, both in wet and dry areas, but the amount of overlap was greater in the wet areas. So this sort of indicated to us that the use of comparing DNA and RNA is going to be a useful relative measure for how much of your community is alive or barely so, and sort of able to pick up. So another way to look at whether microbes are alive, aside from genetic material, is looking at what are the chemicals that they are producing. So we started to look more aggressively at microbial volatile organic compounds. And we know that the smells that microbes emit can be an indication of what's going on. So we know that compost smells. We know that when it rains, you have this nice smell of geosmin in the soil. Those are from bacteria. And we can all sort of imagine that walking into that house would not smell good. And all that is sort of an indication of microbial activity. So what we have done is, in collaboration with some engineers and chemists, is look at the microbes on surfaces in homes and the associated chemical signatures that they produce. So we have these, we put out these common materials in homes. So we refer to kitchen coupons and bathroom tiles. These sit out for a month. Then we take them back to the lab and can use different analytical approaches to see what are the microbiological and chemical communities associated with these surfaces. And this was enabled by a new instrumentation provided by the Sloan Foundation, which provides highly time-resolved signatures of the chemicals and to very low detection limits. So that's what we're looking at here is this really highly detailed instrument set up in the garage of the home that was sampling in the home when we studied. And the way that it worked was these removable surfaces could be put in a jar, flushed with zero air, and then we could ask what are the chemicals coming off of these surfaces specifically. So as a broad, just a broad survey of what we found. So the mVOC composition is going to differ. We saw very clearly it differed between the garage air where the instrument happened to be located, the kitchen coupons, and the bathroom tiles. And you could see that sort of the surfaces in the homes looked more similar to each other than to garage air, but that there were some differences between coupons and tiles. So here's the summary of what we found. We looked at two different time periods, comparing samples on the right with controls on the left. And a couple of things to point out is that we saw seasonal differences, so sampling one occurred in the summer, and there was overall much more production of chemicals in the summer months than in the winter months. And that in general, kitchen coupons were much more chemically productive than bathroom tiles. Most of what we saw were derivatives of these long-chain fatty acids. So we saw a lot of short-chain fatty acids and medium-chain fatty acids. And these are general indicators of microbial metabolism. So the 19 most abundant VOCs contributed most of the total VOCs that we observed, and it was these rare VOCs that were separating kitchen coupons from bathroom tiles. And to me, this is particularly noteworthy because this is highly paralleled with what we see in microbes, where that there's there's a limited number of taxa that are comprising most of the communities, and a lot of these differences in substrates are driven by subtle changes in rare taxa, not the dominant ones. And so a similar pattern seemed to be happening with chemistry. Now, one of the questions is, what we observe chemically on these surfaces, are they microbial derived? And our sampling system, because of the nature of sampling, it's destructive, we couldn't definitively answer that. But we had several indications that what we were observing were microbial. So this is just a microscopic image of what a kitchen coupon looks like at 10x, and this is stained for genetic material cytogreen. And you can see that there looks to just be sort of general dried material, something splashed on it and dried and leaves this trail. And when you look very closely at one of these ribbons, you see that it's actually a ribbon of rod-shaped bacteria. So you can actually see that these surfaces are colonized with an appreciable amount of microbes, indicating that they're likely doing something that we could tell chemically. Also, we correlated the amount of VOCs that we observed with an estimate of the biomass from each of these surfaces. And what we see is that there's a nice correlation. As the biomass increases, so does the quantity of the volatile organic compounds that are coming off. And again, we looked at 2 different time points, so we were comparing summer and winter circling times, and those have different slopes, indicating that there's slightly different dynamics, but overall, as you get more microbes, you get more chemicals. We did some modeling to see if the quantities that we observe of chemicals coming off these surfaces, how those would contribute to house-level mVOCs. And interestingly, overall, the amount of mVOC production from the bathroom stalls is less. The amount of bathroom stall material or surface area in the home is greater, so that ends up being a greater contribution or predicted contribution to the total VOCs. And what we see is that these surfaces could contribute something like 24 parts per billion to indoor VOC contributions. And previous sort of household surveys have indicated that the range of concentration of VOCs in homes is about 100 to 200 micrograms per cubic meter. So we're saying that these surfaces do have a potential to actually be contributing to a lot of the VOCs that we see in homes. Now, in addition to the volatile chemicals, we also looked at the soluble chemicals. And to do this, we compared the chemicals that we observed on the tiles that were left out in homes with the chemicals that were observed when we grew the microbes from those materials and saw what those were capable of producing, just to get a sense of is it likely that the chemicals we observed on these surfaces are deriving, again, from the microbes. And there's about 30% of the microbes were on both, indicating that there's a potential to come from microbes. We see a lot of recent examples of looking at chemicals and microbes. You see general tracers of microbial metabolism, but then you also see a lot of chemicals coming from personal hygiene products. And only 4% of the spectra of chemicals observed had a known name in the database. So highlighting that a lot of the chemicals that are in our homes, we can't even put a name on. So looking forward, a lot of these techniques that we've used in non-water damaged homes and that we've used to characterize what is sort of a typical home are going to be really important when we're going into problems homes and trying to identify how these homes affect health. RNA may be a useful way to get at some of these live dead questions, particularly for indoor environments where the nature of the sampling and low biomass are problematic. And volatile chemicals may be useful. They They may be very general indicators of viability. They, in a lot of ways, they're easier than, and quicker than genetic-based markers. They might not tell you if it's alive, but they might give you a sense of what they're doing. But a lot of the basic aspects of mVOC production in these microbes in these settings is unknown. So with that, I am done. Thanks.
Do we have a question for Rachel? Any questions? Okay, so that was another very nice talk. The point about chemicals that we don't know, the large number of chemicals, we found very similar results when we've done non-targeted screening for articles and products in the home. So this is going to be a big area.